Question 156. How many of the following are correct with reference to cellulose? Cellulose, you all know that is a polymer which is made up of glucose as monomeric unit. In first statement it is given it is a polymer of glucose. This is correct regarding cellulose. Statement 2 is given it has branch chain. This is incorrect regarding cellulose since it has unbranched structure. So this cannot be an answer. Option 3, that is, it is a homopolymer, means it is made up of only one type of monomeric unit, that is glucose. So this is true regarding cellulose. Option 4, it occurs in plant cell. You know that plant cell wall is made up of cellulose. So this is true regarding cellulose. Statement 5, cotton fiber and paper contain cellulose. This is also correct. Statement 6, its monomeric unit is N-acetyl glucosamine. This statement is incorrect regarding cellulose because its monomeric unit is glucose. N-acetyl glucosamine is monomeric unit of chitin. Then there is 7th statement. It gives blue-black color with iodine. This statement is incorrect regarding cellulose because it lacks complex helices. It lacks complex helices which can hold iodine molecule. That's why it gives negative iodine test. Blue-black color with iodine is produced by starch molecule, not by cellulose. So among statements, 4 statements are correct regarding cellulose, making option 4 as the answer for our question. Question 157. Mark the incorrect match. Option first, that is ribozymes, and it is matched with nucleic acid with catalytic power. You all know that RNA molecule having catalytic activity are known as ribozymes. So they are basically here RNA molecules with catalytic power. So this is a correct match here. Option 2, that is fibroin. Fibroin, it is matched with protein in silk with secondary level of organization. This is a correct match because fibroin protein is present in silk and it has beta pleated structure. So this is a correct match here. Option 3, that is insulin. It is a polysaccharide hormone whose tertiary structure determines its specificity. This is an incorrect match here because insulin is a proteinaceous hormone, not polysaccharide hormone. This is an incorrect match here. Option 4, that is sucrose. It is matched with non-reducing sugar with glycosidic bond. You all know that non-reducing sugar, they lacks free aldehyde or ketone group. So non-reducing sugar, they lacks free aldehyde or ketone group, because of which they are unable to reduce cupric ion to cuprous ion, which are present in Benedict solution. And non-reducing sugar, you all know that they give negative Benedict and Felling's test. So in sucrose, which is a disaccharide and it is made up of glucose and fructose, the aldehyde group of glucose, which is present at first carbon, and the ketone group of fructose, which is present at second carbon, both of these aldehyde and ketone group are involved in formation of glycosidic bond. So sucrose, it lacks free aldehyde and ketone group, so it is a non-reducing sugar with glycosidic bond. So it is a correct match here. So among the given option, option 3 is an incorrect match and is also the answer for our question. Question 158. A new amino acid in a polypeptide chain is added at. So if we consider the polypeptide chain in a form of a line, then the left end of the chain is represented by the first amino acid, means this amino acid is the first amino acid and is also known as end terminal of the chain. So this is the left end is represented by first amino acid and is also known as end terminal amino acid, while the right end is represented by last amino acid and is also known as C-terminal amino acid. And a new amino acid in a polypeptide chain is added at C-terminal of a chain, means after this last amino acid. So making option 4 as the answer for our question. So let's proceed to our next question, that is question 159. This says find the correct match. In column 1 there is given group, and in column 2 there are given various examples of secondary metabolite. So pigment here matches with anthocyanin, so A here matches with 5th. Alkaloid, an example of alkaloid is morphine, so B with 1st. Toxin, example of toxin is ricin. And D, that is polymer, and rubber is a polymer, so D here matches with 3rd. So the answer for this question is option 4. Question 160 says Emil Fischer put forward. So you all know that Emil Fischer put forward one of the famous hypotheses to explain the mechanism of enzyme action, and he put forward lock and key hypothesis. As you can see in the diagram, according to the lock and key hypothesis, the active site of enzyme has geometrical shape complementary to that of the substrate, like a lock and key. As a result, the substrate it can easily bind with the active site of enzyme. Substrate का shape जो है active site से complementary है, तो सिर्फ substrate ही active site से bind कर सकता है. And once a substrate bind with the active site of enzyme, it forms enzyme substrate complex, which result in the change in the structure of substrate and leads to the formation of product. But the shape of product is no longer complementary to that of the active site of enzyme. So product is released and enzyme is free to bind with another molecule of substrate. So Emil Fischer put forward lock and key hypothesis, making option 2 as the answer for our question. Talking about the rest of the option first, that is RNA model. You know that there are 3 kinds of RNA molecule, that is messenger RNA, rRNA, tRNA, and these 3 different types of molecules are discovered by various scientists. Like mRNA is discovered by Jacob and Monod, and tRNA is discovered by Paul Zamachnik, not by Emil Fischer. So option first cannot be an answer. Option 3, that is induced fit theory. Induced theory is another hypothesis to explain the mechanism of enzyme action, and this theory was put forward by Koshland. Lock and key hypothesis के हिसाब से क्या था कि active site and substrate they have geometrical shape complementary to each other, but according to induced fit model, the active site of enzyme and substrate they do not necessarily have complementary shape. The active site of enzyme is induced to change its shape so that substrate can easily bind to it. मतलब इनका starting में ही complementary shape नहीं होता है, but once the substrate start binding, the active site of enzyme it induced to change its shape so that it binds it's completely with the substrate. Induced fit model is more accepted as compared to lock and key model because there are various enzymes which can bind with more than one substrate, and those substrate differ in structure. अगर structure differ होगा तो lock and key model तो apply ही नहीं हो सकता, because according to lock and key model the active site of enzyme have the complementary shape like that of the substrate, फिर तो कोई दूसरा substrate उससे bind ही नहीं कर सकता. But according to induced fit model, the active site of enzyme it do not have a proper shape, but the active site is induced to assume a shape that is complementary to that of substrate. So induced fit model can explain all those enzymes which can bind with more than one substrate. That's why induced fit model is more accepted as compared to that of lock and key hypothesis. Option 4, that is DNA model, and you know that DNA model is given by Watson and Crick. So this cannot be an answer. So let's proceed to our next question.